Hello, my name is Yiming. Together with my teammate Ziyuan and Yihan, our topic is generative learning, TCR, and antigenic peptide interactions via pre-trained encoder-decoder transformer. Let me introduce our work first. So what is these questions? T cells use TCRs to recognize antigenic peptide, which can create immune response that can defend the virus. So, if we can design the TCR sequence or peptide sequence, this immune response is controllable by human, which means this problem is very important in adaptive immunotherapy. Deep learning model can capture high-dimensional patterns between the sequence, so we use this sequence-to-sequence transformer to trying to solve this problem. But this approach has several challenges. First is the complexity of TCR and peptide interactions itself. The second challenge is the limitation of high-quality peer dataset. The third challenge is the information loss due to the homogeneous target mapping. That means one peptide could be corresponding to many TCR, or one TCR could be corresponding to many peptides. Our approach is trying to use transformer to learn this pattern, and we validate our model on experiment validation. The research field related to our topic include language model and generative AI for protein design. So our method basically include two steps. Step one is training. So this training strategy is teacher forcing. That means during training process, we doesn't rely on time step, and we just use next token prediction of the reference sequence. The step two is inference, and the inference process is autoregressively. That means in prediction process, we predict next token rely on the previous prediction that relies on the time step. That means autoregressive model. So using this training method, we validate it on two problems. One is TCR to peptide prediction. Another is peptide to TCR prediction. And our baseline is basically include recurrent neural network like LSTM, GRU, and bidirectional LSTM, and convolutional neural network, which is one-dimensional. And also the transformer architecture, which include BART and T5. So here is our main result. This result indicates that T5 performs best, and because it has the highest BLEU score and F1 score. And pre-trained model outperform non-pre-trained model ones. Even the pre-trained weight is on the nature language, not on the protein sequence. And also, the small model, although it have high efficiency, but it's not capable to learn this interaction. And for peptide to TCR, this task is the conclusion is very similar to TCR to peptide. We also do some ablation study. Specifically, we studied it on the decoding strategy of our model, and also the model scale. We try to scale up model and with the condition of with pre-trained weight or without pre-trained weight, and we find that pre-trained weight really helps, and scale up models will also helps. And we also do the ablation on the data amount. We use 25%, 50%, 75%, and 100% of training data to show the accuracy of prediction. And we find that for TCR to peptide prediction, scaling up the data is useful. And for peptide to TCR prediction, it shows some unpredictable and unstable relation. And finally, we do a sequence distributional analysis. First is the impact of model scale. We use T5 small, base, and large. We scale up the model and we find that the prediction distribution is more diverse while the model scaling up and more similar to the wild type protein. And here is our presentation method. We show every position of peptide and draw this conclusion. So our conclusion is use model architecture, encoder decoder, and our model based on T5, and pre-trained weight helps, and the larger models and larger data also helps. And we find that the best decoding strategy is beam search with beam is equal to 2. And in the future direction, we can try to explore with larger dataset, and we can maybe have some vet experiment validation, and also some practical application in immunotherapy. Thanks for watching.